Hello again. This time we're going to cover how to annotate your finished assembly using Prokka. So right now I'm in the test_assembly, so it was the assembly that you had completed. You might have called it something different, but just make sure you're in the folder now before continuing. And we also have here now that cost report from last time, so that's something we did as a metric. I don't need it for the next step though. What we're going to use is this command, Prokka. We can also take a look at it using -h to see what the usage information is. So that's what I'm doing, is I'm just pulling it up. In this case, what we're dealing with is a mitochondrion, and you can see right here the -- kingdom is what you can use to specify what annotation mode it's going to use. And this is particularly useful for things like when you have to annotate the open reading frames, you might have to use a different genetic code variant. And also, it's going to change the strategy of, I think, Aragorn in terms of what it should be looking for, like 16S, 18S, etc. So here we have different options, and by default, it's bacteria. So a lot of times you don't have to worry about this setting. If you're only ever interested in bacterial stuff, just set it to bacteria. By doing nothing, actually. You don't have to include it. But in this case, it is mitochondria, so we will need to include that. Then there's some other options that aren't terribly useful. In this case, really the defaults carry you all the way through. But we will have to specify an outdoor directory. That's outdoor for our directory that we would like to create to put our output. And really that's about it. So why don't we go ahead and give this a try. And to do that, I do Prokka -- kingdom mitochondria, because that's what we're dealing with. If you're going to be dealing with your samples and they happen to be bacteria, then you don't have to do this step. And then you can also do CPUs, -- CPUs, although it does 8 CPUs by default, which is probably more than enough for what you need to do here. And then I'm going to give it that out directory. Notice that it's -- outdoor instead of -o. So it can be frustrating sometimes because programs might have different options to specify the same thing. You know, like so it might be -o in one program or -- outdoor in this one. So it's unfortunate, but you just have to kind of look through the help file to guess. And I guess I will call this Prokka report to kind of match what we were doing with the cost one. And then we just have to give it at the very end the contigs file. So the one contained in our assembly. And that's about it. I'm going to hit enter and it's going to start working. Now, it might take a while on very large assemblies, so you might want to add no hup. But for this, it should be very fast, hopefully. And it's running a series of tools. So it's running Aragorn, Barnap, BlastP. There are a lot of tools that it's going to use because it's basically a pipeline. It's got these things all lined up that it's going to use to analyze your data and then annotate it. And that's what we're going to see as the finished product. And it will tell you what it's doing in the meantime. And we should be almost done. Oh, there we go. Okay, took longer than expected, but not too bad. So it used about 0.8 minutes, that's fine. All right, so now we can actually check out what it did. I'm doing another ls. You can see the Prokka report right here. So when yours is complete, you should be able to go right in there. I would ignore these. These somehow seem to pop up whenever you do mitochondrial. But the rest of these are the pertinent files. The first thing we can check out is the .txt extension. So they're basically all the same name except they're differing on their extension. And it's the extension that's going to tell you what it's containing. So like I said last time, it's really important to keep track of your extensions. This .txt file is just going to give you a very general description of what it annotated. So it said there was one contig, and those are the bases of all the contigs summarized. And then it's going to show you the tRNAs it found, the CDS it found, and the number of rRNAs, so ribosomal. And now some of the other files. Oh, so there's also a log. So this might be useful if you want to keep track. This log right here might be useful for methodology or something. You can just tuck it away somewhere because it will just tell you what was run. And now for these, this is going to be the amino acid file. So if I take a look inside of here, the FAA file, you'll see that here are some of the annotated proteins from our mitochondrion. And it's got what you expect. All right. Now we can also take a look at the GFF file, although it's kind of, it's actually going to be kind of small because we've only got a single config this time. So what you're going to see right here are the, oh, you know what? I forgot something really important. I just quit out of this. I forgot the -S to make this readable. So you can just go ahead and just modify it with that. So less space -S, and again, we're using the GFF, so they're much better. Now we can see one line per annotation. So then you can kind of see the type of annotation here, the tool that was used to annotate it. Aragon was used to annotate the tRNA. What was used to do the RNA? Barnap. Whoops, so my mistake. Aragon is what's going to be used to annotate the tRNA. So anyway, and over here we have, again, I actually already described this before, what a GFF looks like, but here we have your mitochondrial GFF file, and we can also see the tRNAs. There's a bunch of them, so that's great. So overall, it's looking good. One odd thing, though, about this is it also decides to include the FASTA file in the GFF file. That's not typical. It is something you can do because some programs might want to take it in the same GFF, but I just feel like that's sort of cumbersome because a lot of times you just want to look at the GFF divested from your actual assembly, so I don't understand why it does that, and I haven't found a way to turn it off short of hacking, but there you go. So I'm going to quit out of this. We still have some other files to take a look at. So let's see. We've got the FSA. This one is sort of straightforward. It's just your same assembly, the same contigs. And then it's got some additional information like the genetic code that was used. And this annotation stuff is only changed if you specify it in the settings. And it's not something, when I first saw this, I thought, oh cool, it's going to automatically annotate the species for me on this config. But unfortunately, it doesn't do anything like that. It's just that you can kind of give it the names to have it automatically add to this part. So, you know, kind of disappointing. You can probably just ignore this one in general, the FSA, because you already have a copy of the config somewhere. And we can do a less of the FNA. Same thing, it's just without the annotations. And then we're going to do a less of the FFN. They've got all kinds of very similar sounding ones. Okay, the FFN is more interesting because it's going to be the CDS version of these proteins that we saw in the FAA. And it can also contain RNAs as well. So you're going to get your CDS and your RNAs. So here's an example of a 16S that we found. And it's already all set for you. You could, for instance, grab this sequence and blast it on NCBI to find out maybe what this is closely related to. And then you can just go through here and you can see all of them. And you should remember some of the tools that we've used from last time. Like for instance, we could use a grep-c to count the instances of this. So I could count, oh, I don't know, the FAA to figure out the number of proteins that I have, 10. So that kind of lines up with the CDS report, remember from the .txt file. So those are the primary things that you should worry about. This is the files that you will get back from Prokka. And this is what you're going to be wanting to analyze. There are also some programs, I believe, that will take the GFF file and help you visualize it, but that might be outside the scope of this video. So until next time.